Good afternoon, everyone. Welcome to this week's ATI seminar. My name is Trish Campbell, and first I'd like to acknowledge that the University of Alberta is located on Treaty 6 territory and respects the histories, languages, and cultures of First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. Thank you also to Paladin for their support of the ATI seminar series. And it is with great pleasure that I introduce Dr. Anat Tambur for today's talk. She is the director of the Transplant Immunology Laboratory at the Comprehensive Transplant Center at Northwestern University, Chicago, and is well known to the transplant immunology community. She has served as the president of the American Society for Histocompatibility and Immunogenetics and the American Board for Histocompatibility and Immunogenetics. She is the founder and co-lead of the Sensitization and Transplantation Assessment of Risk STAR initiative, which is a collaborative working group of ASHI and AST. She is currently also the co-chair of the AST Research Network Career Development Grants in the translational section and serves as an associate editor for the AJT Clinical Transplantation and is an editorial board member for additional transplant-related journals. Her research focuses on understanding humoral immune responses in the context of solid organ transplantation, with specific emphasis on HLA-DQ epitopes. Her work has led to more than 130 publications with numerous national and international invited presentations in the field, for which she was recently recognized as the Bernard Amos Distinguished Scientist Award at ASHI last year. She's passionate about educating a new generation of HLA laboratory director and scientists and has trained to date 12 candidates. Clinically, she oversees a busy transplant immunology lab in support of solid organ and stem cell programs. The title of her talk today is Towards Understanding HLA-DQ Immunogenicity. Thank you so much, Anat.
Thank you so much. And let me just get to my presentation. All right. Hopefully everyone sees this well. It's a great pleasure to be here with all of you. I apologize, I'm not there in person, but I'll be happy to have as much discussions as guys would like to have. I will talk about this beautiful molecule here. This is a painting that is in my office, reminding me how interesting the HLA-DQ molecules are. I do not have any relevant conflict of interests. And I thought I'll start with data that maybe some of you have seen before, but I think it's really critical to continuously remind everyone that when we're talking about HLA-DQ molecule, we need to consider the protein on the cell surface that is combined of an alpha and a beta chain. I know a lot of people in the field relate to the DNA because we're talking about two different genes, DQA and DQB, but on the cell surface is one molecule. So I'll show you kind of like how we stumbled on why it's so important to talk about the heterodimer, what might be the clinical implications of this. And then I'll talk a little bit about molecular mismatch analysis in the context of HLA-DQ. And towards the end, I'm going to share some unpublished data that I'm really, really excited about that I think really gives us a different insight of immunogenicity, at least in the context of HLA-DQ. So I'm going to take you about 17, 18 years ago, probably a little after I joined Northwestern, and we had a recipient who needed a kidney transplantation and his brother who stepped up as a potential living donor. At the time, our way to analyze HLA antibodies, that was just as the Luminex started to show up, but in the lab, we had what we call the high definition beads. It's a flow cytometry assay looking at single antigen molecules attached to beads, but analyzed on a flow cytometry. So it wasn't as multiplex. What you see here is one tube that contained all the DQ reagents with one analyte per each of the DQ specificities. And I've highlighted for you here in this red box a strong response against DQ7, as you can see here. This we assume to be a donor-specific antibody because the donor was DQ7 positive. So we projected the cross-match to be strongly positive. However, when we ran the cross-match results, we found that both the T cell and the B cell were completely negative. So that didn't make a lot of sense. And we were trying to figure out what was happening. And as I said, the Luminex was just introduced into the field. So we decided we're going to go ahead and run that Luminex. I apologize for all the emails. I thought I've closed that, but we're going to let that go. So on the Luminex platform, one of the really nice thing about it is that we have multiple analytes per each serologic specificity. DQB0301 equals DQ7 serologically, but the difference between the different beads is the alpha chain, or the A gene that contributed to the alpha chain that comes together as a heterodimer with the DQ beta pair. And as you can see here, we got 4 strongly positive results, and then one of the beads was completely negative. And when we high resolution typed the intended donor for their DQ7 alpha beta genes, we found out that there was DQA0301, DQB0301, which is this top bead that was completely negative. And that really jives very nicely with a negative flow cross-match. And then when we reached out to the vendor of the flow high definition beads, we found out that the alpha chain associated with this DQB0301 is alpha 05, which is present on 3 of the beads of the Luminex. So again, these were concordant results. So it's kind of like true, true, and unrelated, if you will. If you have the right alpha beta combination, you have the right result. So this is when we started thinking about what is really the role of the alpha chain in the DQ heterodimer. So one of the things that we've done in order to get a sense of this is looked at about 100 patients that we already knew that they have DQ antibodies, because we had the flow high definition beads for them, but then we ran them on a Luminex platform, and we also high resolution typed them to know what their own typing. And what we found was really surprising at the time. So I've highlighted here or bolded here 5 beads that can look for DQ7 antibodies. And 2 of them are strongly positive, as you can see here. 3 of them are negative. Now, the patient himself was typed as DQ7, DQB0301, with an alpha chain that is not present here. And in essence, we found that about 1/3 of our patients had antibodies to a DQ molecule that serologically would have been considered to be self. But when we looked at the alpha beta combination, it made a lot of sense. So never an antibody to a self alpha beta heterodimer, but 1/3 of the cases, antibodies to self DQ beta chain when it was in combination, a heterodimer with a non-self alpha. And if that case, then can we have combinations of self alpha chain with different betas that will give us antibodies? So here is one example of a patient that was typed as a DQA0201, and you can see the 5 beads that have DQA0201 in the DQ heterodimer. In this case, the heterodimer was together with a DQ beta 0202. You can see that this bead is completely negative, but when there were other DQ betas associated with this alpha, we could see some positive results, and that happened in about half of the patients. So that really led us to think that the alpha beta heterodimer must be thought of as one entity together. We've done another study where we followed René Duquesnoy's approach to try to identify the epitope. So this is almost 10 years ago. And we had 47 patients where we knew that they had no HLA antibodies prior to transplant. They were transplanted with a DQ mismatch donor. They went ahead and developed de novo donor-specific antibodies. And we've used the approach that the matchmaker had proposed at the time to try and figure out what is the applet that is driving the immune response. So here in this particular case, I'm showing you one patient where this is the DQ molecule, the alpha chain in pink, the beta chain in blue, following the antibody responses, knowing the typing of the recipient and the donor, and using HLA matchmaker. The conclusion was that we're talking about an applet on the DQ beta chain with a particular name that really is not very relevant. What we've done at that point, though, is we used CM3D. This is a free software available from the NIH that if you tell the software what you think is the core of the epitope, then it will draw an area of about 15 angstrom radius. So this is what you're seeing here in yellow. That would be supposedly the footprint of the antibody, or what we refer to as the epitope. And then when you take this area and revert it back to the original colors of the different chain, this is what you're seeing here in the smaller figure, then you see that the epitope is contributed by both portions of the alpha chain, beta chain, actually a portion of the peptide. But again, despite the fact that the applet is on one of the two chains, epitope contains an area that combines both of them. Some more examples from this paper. So here's another example where we have the applet identified to the beta chain, but when we look at the area that is supposed to be the epitope, we see alpha and beta. Two cases where the applet was located on the alpha chain. In this particular case, it's just the applet on the alpha chain. The rest of the epitope was proposed to be mostly on the beta chain. And of the 47 patients that we've studied, only one case we found an applet on the alpha chain, and the epitope or proposed epitope being only on the alpha chain. So again, I will go back to my point that I'm trying to drive for many years now. We really need to look at HLA-DQ molecules as combinations of the alpha and beta, not talk on the alpha a separate entity and the beta as a separate entity. And why is it so important? Because it has a lot of clinical ramifications. Before I want to go there, this is specifically for the HLA folks in the crowd. We know that the class 2 molecule has a very similar structure to the class 1 molecule, and no one considers looking at class 1 molecule based on the different extracellular domains, right? So we have one gene that encodes the alpha 1, alpha 2, and alpha 3 domains, versus the class 2 where we have an A gene encoding the alpha and a B gene encoding the beta. But when we're talking about epitopes of class 1, no one will consider talking about an alpha 1 epitope for this particular molecule, or an alpha 2 epitope for another class 2 molecule, because we're thinking of the gene. When antibodies look at the molecules, they don't see the genes, they see the complete molecule. So the alpha and the beta component of the DQ molecules should be treated just as well as we're doing for class 1, the alpha and beta. Again, must always consider the alpha beta chains together. And just as a shout out to our autoimmunity colleagues, they have been referring to DQ molecules as pairs of alpha beta all along. The nomenclature is a little bit different. So they will talk, for example, of DQ 2.2 or DQ 2.5 with a beta chain or the serological specificity as the first component of the name, and then the alpha chain as the second one. So that would be a DQ alpha 02, DQ beta 02, or DQ alpha 05, DQ beta 05. And we shouldn't be referring to those molecules differently, because, as I said, I do believe it has a very significant role as we're doing our analysis in transplantation. So I want to share an antibody profile of one particular patient, and we've studied many of those, showed many cases like this. In this particular example, you see the single antigen bead assay with the MF5, so some strong antibodies coming up here, and I've highlighted specifically 2 alleles of DQ2, right? And the serological nomenclature is going based on the beta chain. So 2 alleles that are really positive. But here are 3 additional alleles that are DQ2 that are completely negative. So if we have a donor that is DQ2, do we have DSA or not? And this is a really interesting question to answer. And one of the things that we have done at Northwestern is we took a really, I think, relatively large cohort of patients that we had family members that we were able to get the DR-DQ haplotype of them. We high resolution typed them and derived the frequencies per each of the serologic specificities. So here's our DQ2. And just to kind of quickly refer from one figure to the other, these 2 alleles represent about 50% of the DQ2 frequency of the DQ2 alleles. These 3 alleles encompass the other 50%. So again, it's going to be a toss of the coin if we don't have the donor alpha beta information, if we don't relate to the antibodies as alpha beta combinations, we wouldn't be able to tell whether we have donor specific antibodies or not. If we have that information, it's going to be very clear cut to say. So these 2 alleles are here kind of in the blue color. We do have donor specific antibodies to these particular DQ2 alleles. The question, if that is the case, then what will be the impact on CPRA? Because right now, at least in the United States, we're using serology typing to calculate the CPRA. They've introduced just recently some molecular alleles coming in with providing a little bit of different CPRAs for this. This, by the way, was a study from 2013. It's still though extremely relevant. And then how would that impact our ability to identify donor compatibility or determine the presence of DSAs, as I've mentioned before? Since in UNOS, we have only serological specificities. This is what is captured in the SRTR. And again, until recently, very recently, antibodies were captured only at the serological specificities. I had to, not had to, it was a lot of fun to do this, but I resorted to my colleagues in France. Christian Jacquelinet is really the one driving a lot of the analysis here. Danny Glotz was the one who kind of helped glue this group together. And what we've done here is they sent me DNA samples from 2 years worth of effective donors for the Paris area. So we were talking about over 1,000 donors that they had. When I've done the high resolution typing, we were able to derive the frequencies of the different alleles relevant to this population. And then what they had internally was the antibody profile of all the patients that were on the wait list for the Paris area. And what Christian had done, he had calculated CPRA using the UNOS formula, looking at antibodies to A, B, C, DR, and DQ at the serological level, which is the way that it was calculated in UNOS. But then also recalculated it, replacing the DQ antibodies based on the serological typing, based on the frequencies that we've derived and the allelic level information of the antibody. So I'll call this the allelic CPRA. And what you can see here is when we've calculated the allelic CPRA, many of the patients actually got less CPRA points. And I'll focus, for example, on those that received 100% CPRA using the serologic calculation. If you converted this to the allelic level, many of the patients lost their points because they didn't have antibodies to all the alleles in a particular antigen group. And you might say, well, this is not the best thing for my patient. I want them to get as much CPRA points because that will translate to allocation points at least in the US. But the flip side of this is that we are blocking different antigens. We're using unacceptable antigens, but if we will not block those unacceptable antigens, and Christian had done the simulation to do this, then the patients will have more potential compatible donors coming to them. So this is pretty much a mirror image, obviously, of one another, but that would really allow the patients to get more potential donors. The other, I think, important thing that is associated with this is the fairness of the system. So I'm sure you're all familiar with the sliding scale of the CPRA points in the blue line, as you see here. And again, focusing on the patients that get 100% CPRA points, patients that have CPRA of 100 will get about 202 points. And if we look at the difference between the patients that have CPRA of 100 by the serological method versus the one that will qualify to get 100% CPRA by the allelic level, this is just a change for HLA-DQ. You see that there is about a third of the patients that are getting actually an unfair share of these really, really high allocation points that are giving to those patients. And if we're talking about fairness in the system, it needs to go both ways. And I think really the patients that can have more compatible donors shouldn't be competing with those patients that have more antibodies and at the allelic level will have 100% CPRA. So we're trying to talk with UNOS to get to introduce this into the system. I've trying to work with this for many, many years right now. I think that's still a problem. I think you in Canada may have better way of changing the system. And I think it's something that it might be good for you guys to consider. And I'll be happy to talk about this later if you will. Bottom line, I think there's a big impact when we consider DQ as alpha beta pairs on CPRA, on allocation points, and on the ability of finding a compatible donor. So I'm gonna leave this here and move to talk a little bit about molecular mismatch analysis and really what interests me, which is DQ immunogenicity. And I don't think that I need to explain this figure to you guys. I'm sure you're very well familiar with this. This is the first paper coming from Chris Wibb and Peter Nickerson using HLA matchmaker. At the time, they called it epitope matching. I think the language had changed over the time, but really looking at the number of applet mismatch load, whether we're talking about DR or DQ, at patients that develop de novo DSA, showing that there is a very, very strong statistical significance or a higher chance of patients developing DSA if they have higher applet mismatch load. And this group had continued working in this field and really publishing very robust papers. This is just a portion of the papers that they have out there. And many, many groups had followed suit as a result. This paper now is about 10 years ago. So for those of you who are not familiar with how HLA matchmaker works, I thought I'll kind of bring this little slide here. And in reality, the matchmaker is looking at the recipient 2 alleles. I'm looking here specifically at HLA-DQ alleles, and take all the polymorphic amino acids that are present in those alleles. Matchmaker does not look actually at amino acids. It looks at small fragments of amino acids. René Duquesnoy had referred to them as applets. And we have here a universe of all the applets that are unique to these particular alleles. It does the same for the donor alleles in the same locus. And then it kind of compare the donor universe to the recipient universe, highlights those applets that are present in the donor bucket, if you will, and also provide you the enumeration, the applet mismatch load between the donor and the recipient for this locus. So as I said, Peter Nickerson was the one really spearheading this in the clinical arena. Many laboratories or centers had followed suit, and we were not different. We really tried to see what happens at Northwestern when we're doing this analysis. We focus only on DQ, not DR, and we spend a lot of time getting the cleanest cohort as we could from our patients. And you can see our DSA positive, our DSA negative group. But at the end of the day, when I've done the analysis, I was really disappointed. This is per definition, I guess, a significant p-value, but definitely not as significant as we've seen in the group from Manitoba or other groups. And I'm ashamed to say it took me several years to figure out why that was the case, why we're so different from everyone else, until I think I figured it out. When you look at all the papers, the different centers are taking consecutive patients. It makes a lot of sense. This is usually what we do in medicine. When I've constructed my cohort, I actually excluded all the pairs where the donor and the recipient were matched for HLA-DQ. My rationale was, well, if the donor is DQ matched to the patient, immunologically speaking, they're not going to generate antibodies. And if that is the case, then they're not informative, therefore I should exclude them. So here's the original results that I showed you. I put those pairs back into the mix and did the analysis. And now, sure enough, I'm getting this really strong p-value. You can see the positive predictive value, the negative predictive value, whether we're including or excluding the matched donor-recipient pairs. But I think this part of the slide really gives you a lot of information. So we've done the Uden analysis. We got the threshold, which was at 8 applets, a little different from some of the publications, but not that far from it. But you can see that a lot of the patients are at the 0 applet bucket or bin, if you will. And those are the ones that are actually DQ matched with the donor and the recipient, between the donor and the recipient. When you look at the rest of the cohort, there is a high degree of overlap between the DSA positive and DSA negative. And those were the findings that drove me to talk against applet mismatch load as a way to consider the risk for the patients, because based on this, I don't see much difference between the groups. We did look also at the allele level, which I think immunologically is a more appropriate way to look at. An antibody does not recognize a universe of alleles. It recognizes either this allele or the other allele. You can see that the p-value here is definitely statistically significant. It's a little more problematic how we're looking at this, because we're looking at different alleles, but in reality, we need to look at patients. At the end of the day, when we've done the analysis, though, the threshold here is much lower. It's only 5 applets. About a third of the alleles are 0 applet mismatch load. They're actually identical. And still, when you look at to the left of the threshold, so the low molecular mismatch load, if we're excluding the zeros that cannot develop antibodies, among those in this area, 20% of the patients still went ahead and developed de novo DSA despite very low applet mismatch load. So I'm really interested in what's driving those immunologic responses or immunogenic responses. And at the time, I was trying to kind of put it in a cartoon fashion to try and say, not every mismatch should get the same value or the same immunologic role. And just trying to consider this, I said my reasoning was, if we're replacing alanine by glycine or glycine by alanine, we're talking about 2 amino acids that are very similar to each other. So one will assume that will not generate a very big difference on the molecule per se. But if we have only one amino acid mismatch, that would be a tyrosine instead of a glycine. Now we're talking about a very polar amino acid, very bulky amino acid. And if we'll try to squeeze it here instead of the glycine, not only that we have that one amino acid mismatch, but the three-dimensionality of this, we will have to wiggle around all the surrounding amino acids. So even though they will not change per se, their location three-dimensionality would be different, and that can have an impact. So we were really trying to figure out what are the best ways to try and understand immunogenicity. I'm going to share data a little bit about absorption elution, site-directed mutagenesis, how we combine the two together, and then talk a little bit about a unique cohort that I refer to as 2 mismatch 1 DSA. So for those of you who are familiar with what we're doing, you know that we've been doing a lot of dilution studies. And as we look at how antibodies titer, we realize that they travel in very unique patterns. So this is a single antigen bead assay from one patient. You can see the dilutions down here. And you can see that there are different patterns. If I'll put it this way, it might be a little bit easier to do it. These are actually, if you put them all together, you get the same picture. And my reasoning was that there probably is one antibody that recognizes these antigens, and another one that behave very similarly, recognize all of these antigens, et cetera, et cetera. And what will be a way to try to tease those out from the serum, patient serum, so that we can try to study them individually. And the way to do it, obviously, is by absorption elution. Be more than happy to share the protocol. We started with single antigen bead individual single antigen beads that we got from the companies. Very quickly moved to work with cells. I think the three-dimensionality of the HLA molecules on the cells is way more physiologic than what we might be getting from the individual beads. And what we were hoping to get is a situation where if we look at untreated serum, we'll see different responses of the antibody. We'll incubate it with a particular target. The target will absorb the antibody, so we'll see nothing in the single antigen bead assay. And then we can elute the antibodies from the target and see something that will be very similar to where we started. Well, I showed you before a lot of dilution patterns. That means that our patients are mostly polyclonal. Very, very few patients will have only a monoclonal antibody. So we started getting a lot of results like this. We're still teasing through this. I think we can get really interesting information from it. But for the time being, I'm going to leave it at that and not share much more until we're very comfortable with what we're seeing. So we can do things with absorption elution, but using a patient serum for the time being is quite difficult. What we can do is we can try to work with human monoclonal antibodies, and I'll show you what we got from our colleagues in Leiden. But for this, we needed to have specific cells where we wanted to try and affect a portion of the epitope. So we've used CRISPR-Cas9 to generate a mutation in a particular allele of DQ7. This is our wild type. This is the mutated molecule. And you can see that the difference between the two is really not that very high, but it's obviously noticeable. And we use these cells at the beginning to look at binding of commercial, so those are not human, commercial monoclonal antibodies. So in blue, you see the binding to the wild type, and in greenish yellow, the binding to the mutated cell. And you can see that the binding is completely abrogated. And we reason that the antibody here must recognize or the mutated area is really critical for the recognition. So the epitope must be somewhere in this thereabouts of the molecule. But we use two additional monoclonal antibodies, again, commercial non-human monoclonal antibodies, and those barely showed any difference between the wild type and the mutated cell. Again, reasoning that the epitope is not affected much by our mutation. It must be a different region of the molecule. So now can we take human monoclonal antibodies? And this is the example that I'm going to share with you. We got this from the group in Leiden. This is an antibody that a mother developed against her child. You can see the amino acids that are mismatched. We used HLA matchmaker to try to assume what is the applet that is driving this, or in other words, what René Duquesnoy and many in the field refer to as the functional epitope. One of the rationale of looking at those applets is to say that, okay, well, we defined what is the relevant applet for this particular binding. This is the important thing. This is really what we care about, because an applet can be considered as a functional epitope. And this is in difference to what, again, the field refers to as the structural epitope. So the functional epitope will be this particular applet. This is our swag cell, and this is position E45 that you see here. Matchmaker defined this as the functional epitope. And again, using the CN3D software, we can try to assume what is the footprint of the overall epitope, or the structural epitope as it refers to. Since this is an antibody against our swag, we can use then the wild type and the mutated cell, and you can see where the area that we've mutated. So this is the wild type. This is where the mutation is. It's not close to the quote unquote functional epitope. It's the very edge of the structural epitope. Yet when we use this monoclonal antibody, you can see the beautiful binding to the wild type, and then we can elute the antibody out of it. But the binding is completely abrogated by a mutation from an area that is farther from the functional epitope, right? We have no absorption here, not whatsoever. And in my mind, this says that it's not really just the functional epitope that is relevant for our binding. We need to understand what is the whole footprint of the antibody to be able to make more claims about where we're going with this. All of this aside, I think really interesting stories, but it really doesn't tell us much about immunogenicity. Studying immunogenicity is quite complicated. I've listed a few reasons, but I think we have a wonderful cohort that does give us some more insight. I'm talking about a reductionist approach that we've used because we used a sub-cohort that I think provides additional layer of information. So if we have a scenario where we have a recipient with two different DQ antigens, and he received a donor that is two mismatch DQ, so a green and a blue mismatches with the recipient can develop antibodies to both of those DSAs. But in about 25% of the time, the recipient will develop antibodies just to one of the mismatches and not the other, generating in a single patient test tube a situation where we can distinguish between a more immunogenic mismatch and a less immunogenic mismatch, because this one in this patient that all the stars align and he was able to develop an alloimmune, a humoral alloimmune response, he still did not develop an alloimmune response to this mismatch, therefore probably less immunogenic. We can interrogate this using the same approach that the matchmaker is doing, so comparing the recipient with the donor along the blue arrow, but we add the layer of adding the comparison between the more immunogenic allele and the less immunogenic allele. And in a cartoon way, we're using a recipient who's homozygote here just to make it a little bit easier. We can look at mismatches that are unique to the non-DSA allele and say, this mismatch is lower immunogenicity. We can have mismatches that are shared between the non-DSA allele and the DSA allele. Here they must be low immunogenicity, therefore here they're probably low immunogenicity. So again, we have second layer of mismatches that are low immunogenicity, and then we're left with those mismatches that are unique to the DSA allele. Those must have higher immunogenicity. So if we take it to individual patients, each composition here is one donor recipient pair. Each letter is a polymorphic amino acid. Top two lines are the recipient, bottom two lines are the donor. In red, you see the DSA allele, the one below is not. Right. So again, just reverting to the colors that we talked about, in yellow, we have mismatches that are unique to the DSA allele. In green, we have mismatches that are unique to the non-DSA allele. And in blue, we have mismatches that are shared between the DSA allele and non-DSA allele, therefore not very highly immunogenic. So if we look at the DSA allele and we say we have 26 mismatches, we really have only 12 mismatches that are unique to the DSA allele. The rest of them should not get the same value. So if we're doing an enumeration, we should at least consider two immunogenic values in this particular case. And this is an extreme case. We have 37 mismatches here for the DSA allele. Only 4 of them are yellow. Only 4 of them are unique. And in this particular case, we actually have more green mismatches, lower immunogenicity compared to the yellow mismatches. So overall, excuse me, I really don't think it's the numbers that are driving those responses. What can we learn from this cohort? And really from here on, actually the previous slide, it's not published data, but I think it's important that people will start thinking about this. Here's our two mismatch 1 DSA cohort comparing the recipient versus the donor DSA allele versus the donor non-DSA allele. I spent a chunk of the COVID time looking at trying to find commonalities between all the DSA alleles, looking at locations, charges, polarities, specific amino acids. And one morning I woke up and I said, you're the one who is saying an antibody doesn't see an amino acid. So why are you focusing on a particular amino acid? Maybe I'm losing the forest for the trees. Maybe I need to take a step back and look a little bit farther away. So one thing, yes, we did find one amino acid that whenever it was present in the donor, in 18 of the cases that I'm showing you here, it was on the DSA allele. But more importantly, this amino acid is actually part of a particular DQ alpha, regardless of the beta in the heterodimer, and this is alpha 05. And I hope my HLA colleague are saying, alpha 05, because when we're in the lab and we're checking for donor-specific antibodies, we're seeing 2 different antibodies coming in really, really high frequencies. And if you're doing titers, you probably see them in dilutions, you probably see them in really, really high titers. One of them is an allele of DQ7. So the beta is the same, and I'm showing you 2 different alpha in those 2 different heterodimers. If the donor is this particular alpha beta combination, it's coming up a lot of the times as a DSA. And the other case is DQ2. All of you are now experts. You see the different alleles of DQ2, and sometimes we have part of them positive and part of them negative. In reality, we can even have a recipient who's DQ2 developing antibodies to this other DQ2 allele, almost always when it's associated with the alpha 05. So this is the alpha 05 that we saw coming up at the 2 mismatch 1 DSA cohort. And working with Vasilis Kosmoliapsis, I wanted to see what's unique about these particular heterodimers. So first, we generated the electrostatic mismatch score using the physicochemical properties of the molecules within the serologic specificities. The scale for this is 0 to 0.6. So this is a relatively high EMS score. And mind you, it's very similar between those 2 groups. But what was more interesting is that when we compare the EMS score between the 2 alleles that have the alpha 05, so these 2 heterodimers, the EMS score is actually lower. So at this point, I'm thinking we really need to look at the alpha beta combinations. I've been seeing this for a very long time. I don't think that we need to have high resolution for this. Single field probably will be enough. What will be the frequency of those heterodimers with the alpha 05? in my 2 mismatch 1 DSA cohort. So this is looking just at the DSA alleles, those that drove the immune response, compared to what we might be seeing in a control population. So we took kidney donors from the OPTN and looked at the frequency there, and you can see that the p-value is really, really strong. We also had the ability to look at another cohort. This is coming from the International Histocompatibility Workshop, excuse me, where I'm overseeing the DQ component. The control here is a little different. This population is almost exclusively Caucasian, so I took the right controls for this. And again, you see that about half of the DSA alleles within the 2 mismatch 1 DSA cohort are this heterodimers with alpha 5, very, very similar p-value. So can we learn from this to the whole cohort, because maybe the 2 mismatch 1 DSA cohort is a unique cohort, looking back at the overall de novo DSA cohort, and I'm focusing here on the Northwestern population. This is the frequency among the OPTN donors, and sorry guys, I see low battery here, and I'm not sure why that is the case. Hopefully there will be no problems here. It is connected. Then you can see a very high increase of your alpha 5 heterodimers in the population that developed de novo DSA, and lower frequency in those that did not develop DSA. But I think what is more important is that the patients that developed, that had the ability, the stars aligned to develop de novo DSA, and they had an alpha 5 mismatch, 98% of the time that was associated with de novo DSA, and very similar frequencies are coming from the international workshop cohort. So we talked about the alpha beta combination. We talked about the electrostatic mismatch score, and I've asked Vasilis to generate for us a heat map of all the different alleles. How would they compare EMS score-wise? You can see here the scale that I told you before, and I paused for just a minute. I won't have the time to go through this, but there is a beautiful paper by Effie Petersdorf that came just a few months ago, looking at hematopoietic stem cell transplantation, looking at HLA-DQ, and what Effie showed at the time. Sorry, I think I figured out the problem here. I just want to make sure that my computer won't not die as I'm talking with you. Okay, I think I fixed this. What she was able to do is to go back to data that we know from the 90s. And at the time, it was Jerry Nippon who showed that HLA-DQ molecules can be divided into 2 major families. He referred to them as G1, G2. Effie showed that this is really important for stem cell transplantation. I think this is really important for solid organ transplantation just as well. So here are the G1 and G2 groups. You can see that the G2 group is relatively homogeneous with relatively low electrostatic mismatch scores, and with very big difference between the G1 and the G2 groups, EMS 3D wise. When we look at the G1 group, this is much more heterogeneous group. It is partly lower EMS scores associated based on the serology, but not always this is the case. And you can see that within this group, we have the highest EMS scores, like the really bright yellows that we're seeing here. So we need to figure out what's going on here. One way to try to do this is to think about the evolution, or trying to get the phylogenetic tree that is associated with different DQ molecules. I think this is the first time that someone generated a phylogenetic tree looking at the alpha betas together. We've done this together with Jérôme Luzon, who's an evolutionary biologist. And you can see that the very first branching of those DQ alleles separate G1 and G2. And a couple of weeks ago, I found this beautiful paper, nothing to do with transplantation, showing that these branches probably have evolved independently over 40 million years ago. So I really think that it's the evolutionarily divergence between those molecules that is associated with the immunogenicity. If we'll take it back to the two mismatch 1 DSA cohort, I showed you that more than half of the times the DSA allele had the alpha 5. I think this is the most immunogenic mismatch. About another quarter of the patients were homozygote to either G1 and G2 or G2, sorry, homozygote to either this or that. The donors were heterozygote. So the patient developed antibodies to the mismatch that is across a G barrier, right? If they were G1, then they will develop antibodies to the G2, and non-DSA was also G1. I'm not going to talk about transdimers. Happy to talk about with my HLA people. But 20% of the recipients themselves were heterozygote for G1, G2. I think we need to understand the differences here. I showed you the heterogeneity on the EMS score. So I think we're just step one into trying to get better understanding of what's happening there. But again, our working hypothesis is immunogenicity is dependent or evolving together with the evolutionary divergence. I want to give a shout out to one of you guys. This is Catherine Tinkham, just to remind you, about 5 years ago. She looked at heart and lung transplant recipients. She was looking for an epitope, but in reality, she did show a very high frequency of heterodimers with alpha 05. So that makes me feel quite better. And we're trying to work on other cohorts right now. We absolutely need to understand the divergence within G1. And then we need to learn how to deal with patients that are heterozygote G1, G2. So baby steps looking into trying to decipher immunogenicity. I want to recognize my inspiration, my collaborators, and the beautiful HLA-DQ molecule. And I'll stop and take questions.
Thank you, Anat. That was fantastic talk. What a huge body of work. Phenomenal. If people can either raise their hands or put questions in the chat. Maybe I'll start. You know, this, you talked about the allocation with CPRA points in the US system. The Canadian system's a little bit different in that you're eligible for national sharing if your CPRA is over 95. And currently we treat allele-specific different, so they're almost under-recognized how difficult it is to access them because we don't give them any points for allele-specifics, we label them. But Rob has been working with CBS on an allele level calculator, which we hope to introduce, which I think will try and even that playing field along to try and do that. Are there any other questions from the audience?
Bill
has his hand up.
Very nice and almost inspiring talk. And the
Thank you.
problems of immunogenicity have not properly been approached as a very important basic science and clinical science program. It should be getting more investment, and I hope that people are lining up to help you with this problem. There's another aspect, of course, of DQ immunogenicity, and that is how it interacts with the T cell receptor. And the immunogenicity of HLA antigens is often thought in only one dimension, the antibody dimension. But the T cell dimension is also probably very important, for example, in celiac disease. And exactly how, one of the things about the DQ is that, as I recall from the crystal structure, the dimer can dimerize. And in other words, in order for it to trigger a receptor on another cell, and this could include the FC receptor once it's bound antibody, it may form higher order structures. And I wonder if this is part of the issue of DQ. Does it have any special way? It's a very important molecule in all of immunology. Why is it so important, and how does it trigger T cells? Why does it trigger T cells so well? Look at all the associations of disease with DQ. And I wish we knew more about how it triggers T cells, and we could visualize the multimers of these critical interactions in all of clinical immunology and transplantation?
This is a fantastic question, Phil. I'm eager to have people who are willing to think with me. I think studying DQ is really, really difficult because 90% of our class 2 molecules on a cell surface is HLA-DR. Trying to understand what DQ is doing is very, very difficult. And most of the reagents that we have are looking at HLA-DR. So I'm very fortunate to have a collaboration with a group in Germany that are helping me with generating CRISPR knockouts. I've shown a little bit of this, but we actually have cells that we've knocked out completely the B genes, all the B genes, so DRB, DQB, DPB, and now we're knocking in individual genes. So I have cells that are decorated now just with DQ molecules. They have the full machinery to have a functional molecule. They still have class 1, but they don't have DR and they don't have DP, and vice versa. I have those that have just DR or just DP, and I think those are the best tools, and this is exactly what we're doing right now. Trying to understand is there a difference between T cells that recognize DR and T cells that recognize DQ, because now I can kind of separate them apart. But we're just at the very beginning, and I think this is an untapped area, and I would love if you have ideas to continue, those studies.
Well, should matching take into account the T cell immunogenicity and the B cell immunogenicity for DQ? Should we have algorithms? We have no idea really about how to predict this. Remember, to make IgG, there's a heavy requirement for T help.
Absolutely.
So it'd be interesting to know that we could actually take that knowledge and build something clinical with it.
Yeah, totally. Give me a few more years. I'm on it.
Anne, you've got your question up?
Thanks, Anat. That was awesome. I learned something every single time I get to hear you speak. I really appreciate it. I have many questions, but I guess building on Phil's question too, I mean, would you ever think about incorporating Pirsch into, and I apologize if you talked about this, I had to step away for a clinical call kind of in the middle, but into this sort of algorithm? We're not going to be bored in HLA. We're going to be able to have all of these things to do that. And then also, I don't know if, again, maybe you spoke about it when I started away, but like cis and trans configurations of alpha beta, and if that's something we need to think about more when we're doing analysis or research in this area. Thanks again.
Great. So no, I'm really stepping away from using all the molecular mismatch load tools. I think it's misleading. I do think that between the G1 and the G2 evolutionary group, if you will, there is a high amino acid difference. But if you take, and apologies for the non-HLA people, if you take 0501, 0301, and 0301, 0301, the difference in the number of amino acids is not high, but you have a lot of antibodies generating there, right? So I don't think it's the number of mismatches. I think it's something fundamental in the how evolutionarily different those molecules are. So I really think we need to look for different tools. I'm certain that we need many, many more cohort. I think the 2-mismatch-1 DSA cohort is one that gave me a lot of insight. I was at the NIH on Monday, so hopefully we'll get along the CTOT20 cohort to look into. If you guys have cohorts to share, please.
I see you're muted, but I can see your name.
Sorry. All right. I am so sorry, guys. My computer died on me. I don't understand. I really, really apologize. I don't know at what point we got cut out, but going back to my response to Anne. So yeah, I'm staying away from molecular mismatch load. For the transdimer, I think this is a phenomenal question. We do know that transdimers can form, but they can form within an evolutionary group. So if you think of even physiologically, if you're looking at your patients, you can have alpha 05 associated with beta 02 or beta 03. Both of them are part of the G1 group. You can have DQ alpha is associated with DQ beta 05 or beta 06. They're both part of the G2 group. But if you try to get an alpha from one G group to associate it with beta with another G group, like a transdimer this way, this will give you a hot beat. You don't find them physiologically. So yes, the paper from Effie talks specifically about those transdimers. I do have both within the Northwestern and the international workshop, some patients that can form a transdimer. So one in Northwestern, two at the international workshop. And those are the only outliers that the patient had, that the donor had an alpha 05, and the patient did not develop antibodies to this because the patient was able to generate a transdimer. I know this is confusing for those who are not HLA people. Hopefully it's clear to the HLA people, but it means that an alpha chain from one DQ entity can bind to a beta chain from the other DQ entity. And this way we can actually have up to 4 different DQ molecules on the cell surface. So I don't see the audience right now since I lost the ability to see this on the computer. But if you're listening, it's going to get even more complicated.
We'll warn him. I also want to tell you, Anat, when you froze, before you froze, kind of like this, like as if you were a DQ antigen presenting your head as a peptide. It was awesome. Yeah. So, yeah.
Very clever graphic.
I'm really, really sorry about that, guys.
No worries. It was great. Really fantastic talk. Thank you so much. And thank you to everybody for a great discussion.
Really great. Thanks, Anat.
My pleasure.
Yeah, thanks. You gave me a lot of homework reading to do now.
Next time, Anat, you'll come and visit us in person, hopefully.
Thank you. All right. Take care.
Thanks, everyone. Goodbye.
Bye.